All right, welcome back to the deep dive. We're always talking about these really elegant systems in biology, right? But sometimes I feel like we take the fundamental stuff for granted.
Oh, absolutely.
Like, have you ever really thought about just how animals deal with water and salt?
All the time, actually. Especially since I got this fascinating chapter on osmoregulation and excretion.
Yeah, well, today's the day we're going to dive deep into that very chapter. And I'm really excited to unpack how animals maintain that balance. You know what I mean? It's like you've got creatures in the ocean surrounded by salt water, others in freshwater rivers and lakes, and then you've got the land dwellers like us.
It's mind-boggling when you think about it, right? The sheer variety of environments and the challenges they pose for keeping internal fluids stable. That's osmoregulation in a nutshell, isn't it?
Yeah, it is. And then on top of that, you have to get rid of waste products, which is where excretion comes in.
And what's so cool is how interconnected those two processes are. They work hand in hand to achieve that ultimate goal of homeostasis.
Right. So let's lay the groundwork for our listeners. We're going to start with the basics of osmosis and osmolarity.
Okay.
Then we'll explore the problems animals face in different environments: marine, freshwater, terrestrial, and how they've adapted.
And we can't forget those nitrogenous wastes, right? The different forms they take and the trade-offs involved.
Absolutely. From there, we'll delve into the fascinating diversity of excretory systems, from simple protonephridia to the intricate vertebrate kidney,
With a special focus on the nephron, of course.
the nephron, that tiny workhorse of filtration and reabsorption. And finally, we'll unravel those hormonal control mechanisms that orchestrate the whole process, making sure everything stays balanced. But first, we need to understand the driving forces behind it all: osmosis and osmolarity.
Right, so picture this. You've got two solutions with different concentrations of stuff dissolved in them, separated by a membrane that water can pass through. Osmosis is basically water moving across that membrane to try and even out the concentrations.
So water's always playing the equalizer.
Exactly. It moves from the area where it's more concentrated, meaning fewer solutes, to the area where it's less concentrated, meaning more solids.
And that brings us to osmolarity.
Right. Osmolarity is simply a measure of the total concentration of solutes in a solution. It tells us how much stuff is dissolved in the water, and it's usually expressed in milliosmoles per liter, or osml.
Ah, osml.
Yeah.
That takes me back to my biology classes.
I bet it does. So, for example, human blood has an osmolarity of about 300 mosml,
Okay.
whereas seawater is much saltier, around 1,000 mosml.
Wow, that's a huge difference.
And that difference is exactly why understanding osmolarity is so important for figuring out how water will move between different compartments, like between an animal's body and its environment.
I see. So the greater the difference in osmolarity, the stronger the drive for water to move.
Mhm.
Now, the chapter also introduces these terms: isoosmotic, hyperosmotic, and hypoosmotic.
Right. These terms are all about comparing the osmolarity of two solutions.
Okay.
So if the osmolarities are equal, they're isoosmotic, no net movement of water.
Easy enough.
Now, if one solution has a higher osmolarity, meaning more solutes, it's considered hyperosmotic compared to the other solution, which would then be hyposmotic.
Okay, so hyperosmotic, more solutes, water wants to go in. Hyposmotic in top. Fewer solutes, water wants to go out. Got it. But then there's also isotonic, hypotonic, and hypertonic.
Yeah.
Why all the different terms?
Well, those terms, isotonic, hypotonic, and hypertonic, are specifically talking about how a solution affects the volume of a cell.
Okay.
They consider not only the solute concentration, but also the permeability of the cell membrane to those solutes.
Oh, that makes sense.
So a solution could be isosmotic to a cell, but if some solutes can't cross the membrane, water might still move in or out to try and balance things out.
I see. So it's not just about overall osmolarity. It's also about what can actually cross the membrane and affect the cell's volume.
Exactly. And that's super important when we're thinking about the challenges animals face in different environments.
Okay, so let's dive into those challenges. First up, the ocean. It's this vast expanse of salt water, but it actually poses a big threat of dehydration to the animals living in it.
Right, because for most marine invertebrates, their internal fluids are less salty than the surrounding seawater.
So they're constantly losing water to the ocean.
Yes, but many of them have a clever strategy. They're osmoconformers.
Osmoconformers, meaning they just go with the flow.
Pretty much. Their internal osmolarity matches that of the seawater, so there's no net water movement. Imagine your body being as salty as the ocean. No need to worry about constantly losing water.
Sounds like a pretty chill life.
It does, but it's important to remember that even osmoconformers are regulating certain solutes.
Oh, really?
Yeah, like the Atlantic lobster. It's an osmoconformer, but it has to actively pump out excess magnesium to keep its internal levels lower than those in seawater. So even though their overall osmolarity matches the environment, they're still working to fine-tune the balance of specific ions.
So it's not just about passively conforming, there's still active regulation going on?
Absolutely. Now, when we look at marine vertebrates, things get even more intricate. Bony fish, for example, are osmoregulators, meaning they actively maintain a different internal osmolarity from the seawater. And since they're hypoosmotic to their surroundings,
Meaning less salty.
exactly, they're constantly losing water across their gills and body surface.
So how do they cope with that constant water loss? Do they just drink tons of water?
Well, they do drink seawater, but that introduces another problem, excess salt. So they have to get rid of that excess salt. They have specialized cells in their gills that actively pump out sodium and chloride ions.
So they drink salt water and then pump out the salt.
Pretty much. And their kidneys also play a role by producing very small amounts of highly concentrated urine. They're constantly working to maintain that balance.
It's like a never-ending tug of war with the ocean.
Exactly. Now, sharks have a completely different approach. They're also osmoregulators, but they've evolved to be slightly hyperosmotic to seawater. And they do this by maintaining high levels of urea and trimethylamine oxide TMAO in their tissues.
Urea. That's a waste product, isn't it?
It is, but in sharks, it serves a dual purpose. It contributes to their overall osmolarity, helping to minimize water loss, and the TMAO actually protects their proteins from the potentially harmful effects of high urea concentrations.
So they're using a waste product as an osmoregulatory tool. That's pretty clever.
It's a brilliant adaptation. And because they're slightly hyperosmotic, they slowly gain water through osmosis and don't need to drink seawater. Excess water is excreted in their urine, and they also have a special rectal gland that helps get rid of excess salt.
So sharks have their own unique way of handling the salty seas. Okay, let's switch gears to freshwater environments, where the challenge is the opposite. Too much water, not enough salt.
Right. Freshwater animals are hyperosmotic to their surroundings. Their body fluids are saltier than the surrounding water.
So they're constantly gaining water and losing salts.
Exactly. And to counteract that, they produce large volumes of dilute urine to get rid of excess water, and they actively uptake salts through their gills and from their food.
So it's like a constant battle to keep from becoming waterlogged and losing those essential salts.
It really is. And then you have those amazing creatures that can move between these two drastically different environments, like salmon.
Salmon, yeah. They migrate between freshwater rivers and the salty ocean. How do they manage such a dramatic shift in osmolarity?
They undergo a process called acclimatization. Basically, their bodies remodel their osmoregulatory machinery depending on where they are.
So it's like they have two sets of tools.
In a way, yes. When they're in freshwater, they excrete dilute urine and absorb salt through their gills. But when they migrate to the ocean, their gills switch to excreting salt, and their kidneys produce less urine to conserve water.
That's incredible. And what triggers this switch?
Hormones play a key role. For example, cortisol levels increase when they enter salt water, and this helps activate those salt-secreting cells in their gills.
So cortisol, often associated with stress, is actually crucial for their survival in the ocean.
Exactly. It helps them adapt to the changing osmotic conditions. Now, let's move on to those more extreme environments, like temporary ponds that can completely dry up. Talk about a challenge.
Yeah, how do animals cope with that kind of extreme fluctuation in water availability?
Some aquatic invertebrates have this incredible adaptation called anhydrobiosis, basically, life without water. When their environment dries up, they can lose almost all of their body water and enter a dormant state.
Dormant state, like they just shut down.
Pretty much. Think of tardigrades, those microscopic water bears. They can dehydrate to less than 2% water and survive for years, tolerating extreme temperatures and radiation. And then, when water becomes available again, they rehydrate and spring back to life.
That's mind-blowing. How do they pull that off?
A key player is a sugar called trehalose. It forms a glassy matrix inside their cells, protecting proteins and membranes as they dry out.
Wow, so it's like a built-in cellular shield against dehydration. And speaking of dehydration, that brings us to the ultimate challenge. Life on land, where water loss is a constant threat.
Absolutely. Unlike aquatic animals, terrestrial animals are surrounded by air, which has a much lower water potential than their body fluids, so they're constantly losing water through evaporation.
So how do they prevent themselves from shriveling up like prunes?
They've evolved a whole suite of adaptations. Many have physical barriers to reduce water loss, like the waxy exoskeletons of insects or the keratinized skin of reptiles and mammals.
Like armor against dehydration.
Exactly. They also obtain water through drinking, eating moist foods, and even producing metabolic water as a byproduct of cellular respiration.
Oh, right. That's like a little internal water factory.
It is. And some desert animals, like camels, have taken water conservation to an extreme. They can tolerate large fluctuations in body temperature, reducing the need to sweat, and their kidneys can produce incredibly concentrated urine.
It's amazing how much their physiology is geared towards surviving in those arid environments. And that reminds me of the scientific skills exercise about the sandy inland mouse.
Oh, yes, that was a fascinating case study.
The way their kidneys could concentrate urine depending on water availability really highlighted how adaptable those desert creatures are.
It's a testament to the power of natural selection, shaping their physiology to thrive in such challenging conditions.
Okay, so we've explored the diverse challenges and adaptations related to water balance in different environments. But all this active transport and regulation comes at a cost, right? It takes energy to move all those solutes around.
Absolutely. Maintaining those osmotic gradients is no easy feat. It requires a lot of energy in the form of ATP.
So the greater the difference in osmolarity between an animal and its environment, the more energy it has to expend to maintain its internal balance.
Exactly. And the cost also depends on things like the permeability of their body surface. If it's very permeable, they'll lose more water and salts, meaning more work to keep things stable.
Makes sense. So you want to be as close to equilibrium with your environment as possible to minimize the energy cost.
Precisely. That's why we see adaptations like freshwater mollusks having lower solute concentrations in their hemolymph compared to their marine relatives. It's all about reducing that osmotic difference to conserve energy.
It's like a delicate balancing act between maintaining essential internal conditions and minimizing energy expenditure.
It really is. Now, we've talked a lot about moving water and solutes, but a key player in all of this is the transport epithelia.
Transport epithelia, those specialized cell layers that are all about moving stuff across membranes.
Exactly. They're the workhorses of osmoregulation and excretion. They're found in various places like the gills of fish, the kidneys of vertebrates, and the Malpighian tubules of insects.
And they're often organized into these tubular networks with lots of surface area to maximize exchange.
Yes, that's a common theme. And these cells are packed with specialized proteins, pumps, channels, co-transporters that actively move solutes in specific directions.
So they're like the gatekeepers of what goes in and out of the body fluids.
A perfect analogy. And a classic example of transport epithelia in action is the salt glands of marine birds, like the albatross we talked about earlier.
Ah, the albatross, drinking seawater and thriving.
Right. Those salt glands are located near their eyes or nose, and they use a countercurrent system to actively pump out a highly concentrated salt solution.
So they can drink seawater, get rid of the excess salt, and still stay hydrated.
Exactly. It's a brilliant adaptation that lets them survive in an environment where we'd quickly become dehydrated. Now, shifting gears to waste removal, let's talk about those nitrogenous wastes. Why are they so important, and why do different animals excrete them in different forms?
Yeah, it's all about ammonia, urea, and uric acid, right?
Yes, and the form they excrete depends on a balance between toxicity, solubility, and energy cost.
Let's start with ammonia. It's the most toxic, but it's also very soluble in water.
Exactly. So for aquatic animals, it's the easiest and most energy-efficient option. They just let it diffuse out into the surrounding water where it's quickly diluted.
So problem solved for fish and many invertebrates.
Right. But for land animals, constantly losing water to get rid of ammonia isn't feasible. That's where urea comes in.
Urea, like what mammals excrete.
Yes. It's less toxic than ammonia, so it can be concentrated in the urine, conserving water, but it requires energy to convert ammonia into urea in the liver.
So it's a trade-off.
Yeah.
Less toxic, but more energy intensive.
And then you have uric acid, which is even less toxic and practically insoluble in water.
That's what birds excrete, that white paste, right?
Exactly. It's perfect for water conservation, especially in arid environments and for animals that lay shelled eggs, but it's the most energy expensive to produce.
So it's all about adapting to the environment and balancing those trade-offs. The chapter also mentioned that the amount of nitrogenous waste an animal produces is linked to its energy budget and diet.
Yes. Animals with higher metabolic rates, like endotherms, generally produce more waste. And carnivores, with their protein-rich diets, produce more nitrogenous waste than herbivores.
It's fascinating how all these factors intertwine. Now let's talk about the different excretory systems that have evolved to handle all these waste products.
Right, and there's this amazing diversity of systems, but they all share a common theme. They involve tubular networks.
Tubular networks, like those tiny pipes we talked about earlier.
Exactly. These tubes provide a large surface area for the exchange of water and solutes, allowing for filtration, reabsorption, secretion, and ultimately excretion.
So it's like a multi-step process of refining the body fluids.
Exactly. Now let's look at some specific examples. Flatworms, those simple creatures, have protonephridia. They're networks of dead-end tubules with flame bulbs that filter fluid from the interstitial spaces.
Flame bulbs, those ciliated structures that create suction.
Yes. And the tubules then modify the filtrate, reabsorbing some solids and excreting dilute urine. In freshwater flatworms, it's mainly for osmoregulation, but in parasitic ones, it's more about getting rid of waste.
So even those simple systems show the basic principles of filtration and modification.
Absolutely. Earthworms have a slightly more complex system with metanephridia. These are open-ended tubules that collect fluid directly from the coleolum.
The colon, that fluid-filled body cavity.
Right, and as the fluid travels through the tubule, useful solutes are reabsorbed and the remaining waste is excreted as dilute urine.
So it's like a more direct filtration system.
In a way, yes. Now, insects have a very different system with Malpighian tubules.
Malpighian tubules.
Yeah.
Those blind-ended sacs that extend into the digestive tract.
Yes, and they work by secreting solutes into the tubule lumen, drawing water in by osmosis. Then, the hindgut reabsorbs water and essential ions, leaving behind a concentrated waste product, mostly uric acid, to be excreted with the feces.
That's a fantastic adaptation for conserving water on land.
It really is. And finally, we arrive at the vertebrate kidney, a masterpiece of osmoregulation and excretion.
The kidney, that bean-shaped organ we all know and love.
Exactly. It's packed with millions of nephrons, these microscopic filtering units.
And each nephron has a glomerulus, a Bowman's capsule, those winding tubules, and the collecting duct.
Precisely. It's a complex structure with different regions responsible for specific tasks.
So let's break it down. What happens to the blood as it enters the kidney?
The first step is filtration, which happens in the glomerulus. It's like a tiny sieve where blood pressure forces water and small solutes out of the capillaries and into the Bowman's capsule.
So it's like a squeezing action that creates the initial filtrate.
Exactly. But this filtrate contains not only waste, but also valuable substances like glucose and amino acids.
So the next steps are all about reclaiming those goodies, right?
Yes. Reabsorption is a major function of the nephron, especially in the proximal tubule. This is where the epithelial cells actively pump sodium chloride, glucose, amino acids, and other essential molecules back into the bloodstream.
So it's like a selective retrieval system.
Precisely. Water follows passively by osmosis, helping to concentrate the waste products.
And the loop of Henle plays a role in this concentration process, right?
Yes, the loop of Henle creates a concentration gradient in the renal medulla, which is crucial for producing concentrated urine.
That's where the descending and ascending limbs with their different permeabilities to water and salts come into play, right?
Exactly. The descending limb is permeable to water but not salt, so water moves out by osmosis, concentrating the filtrate.
Okay.
Then the ascending limb is impermeable to water but actively pumps out salt, further contributing to that medullary concentration gradient.
So it's like a two-pronged approach to creating that osmotic gradient.
Precisely. And this gradient is key for the final stage of urine concentration in the collecting duct. As the filtrate moves down the collecting duct, water is drawn out by osmosis, making the urine hyperosmotic to the body fluids.
So it's like a fine-tuning process, squeezing out the last bits of water to conserve it.
Exactly. Now, the distal tubule also plays a role in regulating potassium and sodium chloride concentrations, and it contributes to pH balance by secreting hydrogen ions and reabsorbing bicarbonate.
So each segment of the nephron has its own specific role in refining the filtrate and producing the final urine.
It's a remarkable example of division of labor at the cellular level.
It really is. Now, we've talked about the mammalian kidney in detail, but the chapter also highlights how the vertebrate kidney has adapted to different environments.
Oh, yes. There's incredible diversity in nephron structure and function across different species.
Like those desert mammals with their extra long loops of Henle.
Exactly. Longer loops create a steeper concentration gradient, allowing them to produce even more concentrated urine and conserve more water.
And then you have aquatic mammals with shorter loops, since they don't need to conserve water as much.
Right, and the vampire bat is another fascinating example. It can switch between producing large volumes of dilute urine while feeding to reduce weight for flight, and small volumes of highly concentrated urine when it's resting to conserve water.
That's remarkable flexibility.
It is. Now, birds and other reptiles have some limitations in their ability to concentrate urine compared to mammals.
They primarily rely on excreting uric acid for water conservation. Right.
Exactly. Their kidneys aren't as efficient at concentrating urine, but uric acid excretion allows them to conserve water very effectively.
It's fascinating how different lineages have evolved different strategies to solve the same problem.
It is. And then you have freshwater fish and amphibians, which are constantly battling water influx.
They produce tons of dilute urine and actively absorb salts, right?
Precisely. Their kidneys are packed with cortical nephrons, which are specialized for producing dilute urine, and they rely on other mechanisms like active uptake of salts through their gills or skin to maintain their electrolyte balance.
So it's a multi-pronged approach, not just relying on the kidneys.
Exactly. And marine bony fish have a unique challenge. Their kidneys are primarily involved in excreting divalent ions like magnesium and sulfate.
Because they're drinking seawater and
Right.
constantly taking in those ions.
Precisely. And they rely on specialized chloride cells in their gills to get rid of the monovalent ions like sodium and chloride. It's a fascinating example of how different organs work together to achieve osmoregulation.
It's like a complex interplay of specialized cells and tissues, all working in concert. And all of this intricate dance of water and salts, is also connected to other physiological processes.
Oh, absolutely. Think about plant stomata opening and closing to regulate gas exchange and water loss,
Okay.
or nerve impulse transmission, which relies on ion gradients across cell membranes.
Yeah. Or even bacterial flagella, using ion gradients to power their movement.
It's remarkable how these fundamental principles of ion movement and gradients are woven into so many diverse aspects of life.
It really is. Now, let's zoom back into the kidney and talk about those hormonal control mechanisms that fine-tune its function.
Okay, so urine volume and osmolarity aren't static. They're constantly being adjusted based on the body's needs, and hormones play a key role in this dynamic regulation.
Right. And one of the main players is antidiuretic hormone,
Right.
ADH, also known as vasopressin.
ADH is released from the posterior pituitary gland in response to increased blood osmolarity, which is detected by osmoreceptors in the hypothalamus.
So if the blood becomes too concentrated, the brain sends out a signal to conserve water.
Exactly. And ADH does this by increasing the permeability of the collecting ducts to water.
By inserting those aquaporin channels, those water channels, right?
Yes, more aquaporins mean more water reabsorption, leading to less urine output and more concentrated urine, bringing that blood osmolarity back down.
It's like a negative feedback loop, keeping everything in check.
Precisely. And that's why things like alcohol, which inhibits ADH release, can lead to dehydration because you produce more dilute urine.
Makes sense. And there are also conditions like diabetes insipidus, where there's a problem with ADH production or signaling.
Yes, and that can lead to excessive urine production and constant thirst.
So those hormonal signals are really critical for maintaining that water balance.
Absolutely. Now, another important hormonal system is the renin-angiotensin-aldosterone system, RAAS, which is all about regulating blood volume and pressure.
RAAS, that sounds familiar.
I bet it does. It's a complex system, but essentially, when blood pressure or volume drops, the kidneys release renin, which sets off a cascade of events leading to the production of angiotensin II.
Angiotensin II, that's a powerful vasoconstrictor, right?
It is. It constricts blood vessels, increasing blood pressure. It also stimulates the adrenal glands to release aldosterone.
And aldosterone promotes sodium reabsorption in the kidneys.
Exactly. And water follows sodium, so aldosterone helps increase blood volume and pressure as well. It's a powerful system for maintaining those vital parameters.
And it's why ACE inhibitors, which block a key enzyme in this pathway, are commonly used to treat hypertension.
Exactly. They help lower blood pressure by reducing angiotensin II production. Now, it's important to note that both ADH and the RAAS work together to increase water reabsorption in the kidneys.
So they have a synergistic effect.
Yes, but the RAAS also stimulates sodium reabsorption, which helps maintain the osmolarity of the body fluids. It's a more comprehensive response to low blood volume or pressure.
So it's like a two-pronged attack on the problem.
Exactly. Now, there's another hormone we need to mention. Atrial natriuretic peptide, ANP.
ANP, what's its role?
ANP is released by the heart atria in response to increased blood volume and pressure. It basically opposes the actions of the RAAS.
So it's like a counterbalance.
Precisely. It inhibits renin release, reduces sodium reabsorption, and blocks aldosterone secretion, all of which help lower blood volume and pressure.
So it's like a safety valve, preventing things from getting too high.
Exactly. And finally, we can't forget about thirst, which is also regulated by the brain.
Thirst, that basic drive to drink water.
Yes. Specialized neurons in the hypothalamus detect changes in blood osmolarity and stimulate thirst when we need to replenish our water stores.
So it's all interconnected, this intricate web of hormonal signals and neural pathways, all working together to maintain that crucial balance.
It's a testament to the elegance and complexity of our bodies and the bodies of all animals.
It really is. So we've covered a lot of ground today, from the basics of osmosis and osmolarity to the diverse challenges and adaptations animals face in different environments. And we've seen how the excretory systems have evolved to meet those challenges, culminating in the sophisticated vertebrate kidney.
And we've also explored those intricate hormonal control mechanisms that fine-tune the whole process, ensuring everything stays in balance.
It's been a fascinating journey. For you listening, I hope you've gained a newfound appreciation for the incredible feats of osmoregulation and excretion that are happening inside you right now and in all the creatures around you.
And as you encounter different animals in different environments, think about the challenges they face and the remarkable adaptations they've evolved to maintain that delicate internal equilibrium. And that leads me to a question for you to ponder. How might things like climate change and altered water availability impact these delicate balances in the future? What new challenges and adaptations that we see emerge?
That's a great question to leave our listeners with. Thanks for joining us on this deep dive into the fascinating world of osmoregulation and excretion. It's been a pleasure exploring this with you.
It certainly has. Until next time.
See ya.